Brilliant, thanks for the introduction. Now I'm going to talk about disease suppressive soils. So a subset of what James has been talking about, and I'm really lucky that James has been able to do the hard work and heavy lifting for me and to define all of the terms. But there is something very specific about soil. And that's that we don't really value it very much, and we don't really understand it. And I think this this quote that we owe our entire existence to a 6-inch layer of topsoil, and the fact that it rains, reveals how important soil actually is. Yet it's probably one of the least understood of all of our planet's ecosystems. And disease suppressive soils are really important. We can find them in nature, and they are incredibly powerful agents in terms of moderating pathogenic organisms, or as we call them, diseases, when they infect a crop. So these are some photos taken from research in my lab. This is tomato growing in either a disease conducive soil compared to one that's growing in a disease suppressive soil. Now these soils were collected from the same farm, from within half a kilometre of one another. The soil type is the same, the nutrients are the same, yet if you infect the plants with a foliar pathogen, as we've done it to both of these sets of plants, you can see the plants that grow in a disease conducive soil have been obliterated by the pathogen, and that's in just 48 hours. But when we have a look at the plants that are growing in a disease suppressive soil, we can see that there are no to little signs of that foliar pathogen destroying its host plant, which I think is really phenomenal, and I think it offers some real opportunities, if we can understand it, to begin to control diseases without the use of pesticides. And there are a range of symbiotic, mutualistic symbioses in the soil. I think a lot of you will be familiar with the idea of mycorrhiza. So these are symbioses between 95% of all plants and soil-borne fungi. And here the plant supplies the fungus with carbon in return for nutrients. And that's what we kind of thought the symbiosis did. That's the full extent of it. And hopefully I'll convince you later that there is something much greater and possibly more interesting to this symbiosis. But we also have bacteria in the soil that associate with plant roots, and many tens of thousands of bacterial species occupying the zone around a plant root that we call the rhizosphere. Now, the rhizosphere is the area of the soil around a plant root that is in direct chemical control of that plant. So plants exude lots of compounds through their roots into the soil that have a range of different purposes. And the extent of the limit of these compounds to influence the soil is called the rhizosphere, or that's at least one definition and my definition of the rhizosphere. Now, within the rhizosphere, we can kind of think about it almost as an information superhighway. A plant, which is here represented as the diamond, can send signals out to attract fungi and different types of bacteria, and they can respond to the plant and change how the plant's physiology, the plant's functioning is regulated. And a number of years ago, a colleague, a long-term collaborator of mine, and Urian Ton, came up with an idea about how these different types of organisms within the rhizosphere could influence plant health. And we built a mechanistic model that is simplified here, and if you're interested, this publication provides more detail, where essentially plants send signals out into the soil, and they want to recruit things like fungi that can come along, and when they interact with the plant, they are going to trigger the plant's immune system. Plants have an adaptive immune system that allows them to deal with pathogens, and here these fungi will trigger these plant defenses. But of course, if you're a symbiont, you don't want those defenses to kill you. What you want is to be able to tell the plant you're friendly, and turn off those defenses, which is exactly what mycorrhizal fungi can do. They can produce what we call effectors that tell the plant's immune system to calm down, but not before the plant has sent a signal above ground to warn its above ground tissues that it might be being attacked and to ready themselves for defense. In the process then of sending these signals to the fungus, the mycorrhizal fungus, it influences the chemistry of the root exudates that are produced by the plant. And we know that in some cases there have been observations that mycorrhizal fungi can cause the recruitment of beneficial bacteria into the rhizosphere, and they in turn can influence the host plant's defenses. So we have this multimodal mechanism of how the plant has essentially outsourced much of the regulation of its immune system to the organisms that live in its rhizosphere. And we were able to test this model a few years ago, and we were able to demonstrate strikingly that mycorrhizas can restructure the communities of beneficial bacteria that live in the rhizosphere. And they can also impact on plant health. So here we've got a simple graph where we had artificially triggered the host plant's defenses. The strength of their response depends on how activated or how primed their defenses are by bacteria and fungi in the rhizosphere. And as you can see, AMF means mycorrhiza, PGPR means plant growth promoting rhizobacterium. And when you add them together, the induction of defense is synergistic. So having more of these organisms in your rhizosphere is a good thing. So just in the last 3 minutes of my talk, I'm going to talk about the pathogen that we work on. It's called tomato root mat disease, or Rhizobium radiobacter, and it causes these absolutely ridiculous protrusions, proliferations of roots in their host plants, which are tomatoes. And essentially this means all of the carbon is used to grow roots, and there's nothing left for roots and for shoots and fruits. And one of the big issues is that this problem is intractable. There's no chemical control. And when it gets into a greenhouse, because most of our tomatoes are grown through hydroponics in a greenhouse, it runs riot. So what we've proposed is a novel solution, is doing away with the traditional substrates that you use in hydroponics, which is things like rock wool or coir matting, and actually engineering a substrate which is designed to take advantage of beneficial bacteria. And if you're interested in growing plants in foam, in this case old mattresses, a quick plug for the recent documentary that we have made about our work called The Catastrophe Garden, where we've grown food in a refugee camp in Jordan with Syrian refugees using old mattresses. So our novel solution is to look to the environment, look to these disease suppressive soils in the kind of way that James has mentioned, to harness and isolate those bacteria, and then to assemble communities within artificial substrates that suppress disease. And we've been able to get quite a long way in terms of doing that. what we've done is to harness a bacterial pathogen that attacks the roots as a sentinel, as an indication of whether the plant's immune system is activated. And we've made these bugs glow so we can see them. And we've been able to have a look in different types of soils. We've got a disease-conducive soil, which is the left-hand bar, and the right-hand bar is a disease-suppressive soil. And we've really shown that the disease-suppressive soils can suppress the pathogen, but also we can transfer the microbial communities from the wild into synthetic substrates, and they still suppress the pathogen. Now, I think that's my time. Obviously, these projects are huge and it's impossible to say what everybody has done, but this is what the team looks like. But I think what I really need to do is acknowledge the important people in the team, the postdocs and technicians who've actually done the hard work. Thank you very much.